now that we're here, okay, I'll move forward to the next contestant of Talk Your Thesis. कहाँ गया? हाँ. This is the fourth contestant, okay, jury fourth contestant. This is Shafana Parveen, PhD student at SRM Institute of Science and Technology. She's mentored by Anushila Chatterjee, Head Science Media Center, TFR Hyderabad. Shafana is a genetic engineer who's equally passionate about dancing and making the most of the life, both inside and outside the lab. And इनका आज जो topic she's picked up is plastic. Oh wow, क्या serendipity हुई है? Plastic, the global threat. Can we defeat it? Okay. So with a huge round of applause, yes. Can we guys applause? Can we welcome Shafana Parveen?
Okay. Am I audible? Yeah? Can we start? So, hello everyone. I'm Shafana from SRN. And in the next 300 seconds, let me tell you a story. A story that not just shaped my PhD, but also my lifestyle. But before we start, can we try something fun? We have 5 seconds, just 5 seconds, to spot as many plastic items as you can. Ready? Go. Yes. 1 and stop. How many did you spot? Quite a few, right? Now comes the real question. Where do you think most of these plastics end up? Here. Feels disturbing and hopeless, isn't it? I get it. I felt the same way too. Until something caught my attention. These slimy patches. At first, just like every one of you, I thought, ew, gross. But then, my microbiologist brain kicked in and I realized those could be bacteria. Think about it. Bacteria are single-celled microorganisms, meaning every action they take is related to their survival. In that case, what could this bacteria and the plastic interaction possibly mean? To understand that, let's first get to the very structure of plastic itself. You see, plastic at its core is basically a polymeric structure, just like this pretty necklace, made of repetitive chemical units that are held together by very strong bonds. Now, imagine cutting this necklace. That's precisely what the bacteria does. Good for me. Scientists have already discovered that certain bacteria release some chemicals called enzymes that target these bonds and break them down into smaller units. With this, to my understanding, nature has already started the cleanup process. But as you see, it's happening way too slow. And that's when I asked myself, can we make this process better or faster? And thus began my research. So I just rolled up my sleeves, went straight to the dump yard to collect some samples. To truly understand something, we need to get to the roots, right? So I turned to DNA for answers. With the current advancement in technology, with just DNA, we could get direct information on which bacteria are present, what do they do, how do they function, all in just one go. And most importantly, we get to analyze every possible bacterium in that sample all together at once. So I decided to use this technology to my advantage. And here's how I went with it. First, I analyzed the DNA from the sample as it is to understand the bacteria's natural behavior. This gave me baseline data of the varying levels of enzymes that these bacteria naturally produce. Then, I gave these bacteria a little challenge. I fed them with only plastic, no other nutrients. Eventually, when these bacteria run out of their usual nutrients, they might have to turn to plastic for their survival, right? Maybe. Very curious about their response, I did a final round of DNA analysis. Interestingly, certain enzymes showed a significant increase in their production. Could these be the key players? Maybe, but I had to be more certain, right? So I cross-referenced these highlighted enzymes with the previously reported studies and further narrowed down to the ones that showed similar functional profiles. This approach helped me to identify the key players that were involved in making this into this. Incredible, isn't it? Well, that's not the end of the story. With a little more computational analysis, I tested the interaction between these enzymes and the plastic and picked the best ones. Now, imagine putting these best ones into a single cocktail or even tweaking them a little to improve their performance. That could do wonders, right? These ideas are still a part of my future work. While we continue this exciting research, the real solution lies in our hands. Thank you.
All right, jury. आपके सवाल का time आ चुका है. Please.
Thanks for this exciting presentation. This is
Thank
very
you.
hot topic. All of the jury member looking at me because I'm working on plastic
Yeah,
pollution.
you're a marine microbiologist. I
Yeah.
realized I would get questions from you.
Okay, so we produced like 10 billion tons of plastics until now. So we have 7 billion tons still in the environment. So how many bacteria we need to degrade all of this
How
plastics?
many bacteria? So that's what I was telling. The process, to our understanding, it's happening way too slow. So I believe as scientists, we need to intervene to kind of help our environment or nature to fasten this process up. Like we can look at genetic engineering, like overproducing a certain enzymes that that could help in plastic degradation. So yeah, I'm not telling plastics are bad, like they are doing a tremendous work, like that's why they're just everywhere. But I think we have to manage them better, like even with just avoiding single-use plastic. Apart from that, to deal with the current plastic pollution, science has to intervene to get it kind of ramped up the process. Thank you.
We should trust bacteria.
We should trust bacteria. So I would like to say not all bacteria are good.
Okay.
There are certain notorious ones. So I think that is why this metagenomics approach, the DNA approach, would help because we are not going to grow all of the bad ones also in the in the lab. So we can't blindly trust bacteria. I like just the first talk, she was talking about infection-causing bacteria. So not all bacteria are good. So we just need to pick on the ones that we want.
Thank you.
Thank you.
And now time for the score. Okay, on the count of 3, 2, 1, as you all do. Okay, 3, 2, 1. All right, 3, 4, and 5. Thank you so much,
Thank
Shafana.
you.
Bye-bye.
Bye-bye.